Hello everyone, happy evening, a very good evening. I hope and I believe all of you are doing well. A quick note whether the audio visual is all good. Yes, I hope I am audible and visible, everyone. And welcome to the FAST episode 6, where we are going to continue the genetics part, the molecular biology part. And FAST is basically first aid simplified teaching. So we know that the biochemistry part from first aid is very, very important. And in the last two sessions, one in the yesterday's YouTube live session, one in the today's free live class on an Academy app, we have seen about the DNA structure. We had seen about the genetic code, right? So yes, thank you so much. E-fast it is. And how long will it take to cover the entire first aid? हमारा target पूरा first aid करने का first of all नहीं है. It's more about quality than the quantity that we are focusing on. So of course, it's not about completing the entire thing, but doing the best with the whatever time we have is what is our target. I'll try to do the best that we can. And in the today's session, we are going to learn about the DNA replication. Then we have DNA mutations and the DNA repair, which is very, very important. And this is the timetable that we are following basically. So today you have to complete the biochemistry part. I hope all of you are on it. And the good news is that we have 25% off on all the medical PG subscriptions till 31st of March. And this is applicable for all the courses, including the NEET PG 2024 batch. Just give me a minute. I don't know why this pen is giving a problem. Just a minute. Okay, let's see. I hope it would work fine. And we also have the short INICT course that would be available on the Unacademy app. Like we had the short for NEET PG and FMGE. Now we have INICT specific, focusing on the topics which are like frequently asked in INICT is what we would be doing in the short INICT. And if you take this till 31st of March, you will get it at a price of 3750, and with your a lot of rewards or credits in your account, you get it at approximately like 3.1k, which is very, very reasonable, I would say. So make sure you make the best of this opportunity before 31st of March. And yes, we have had a lot of learners who have got great ranks in the last NEET PG exam. All the students who are planning to appear for the next NEET PG exam, this is the course which is meant for you, the 9-month course. We have started recently on March 28. It's a 9 months course where everything would be covered for the upcoming NEET PG exam. And you can take a combo subscription also that is live and recorded till 31st March. This is available at approximately 18k, right? Now, this is the part which we have covered from first aid already. We have seen the chromatin structure, the various changes, the nucleotides, right, the purine and the pyrimidine synthesis. This part is what we had seen in the today's special class, right? This was covered in the today's special class on the app. So whoever missed that, you can check that out. We will be having next two sessions also in biochemistry based on first aid teaching tomorrow. That is 31st of March. We will have two more sessions. We will have one at 11 a.m. right, and we would have the second one in the afternoon, that is at 2 p.m. right. So we would have both of them 11 a.m. and 2 p.m. Okay. Yes, thank you so much for that. Unacademy Plus, if you are having Megha, you will get the access to the short INICT. Yes, Rekha, it would be available to the Plus students without any extra charges. Right, it's not a different subscription for the Plus students. And this is what we have covered in the special class today. And this is what we have covered in the special class today, including the Lynch syndrome and also the genetic code features is the last that we had covered in the special class today. So anybody who missed that, you can watch it as a recorded class on the Unacademy app. The link has been shared on the Telegram group. The links for the tomorrow classes, the 11 a.m. and 2 p.m., where you would see the rest of biochemistry, will be shared in the Telegram group after this session, right? So now starting with the DNA replication and focusing on the points that are important. Just give me a minute. The pen is giving some problem. I hope it works fine at least till the session. ठीक है. Sure. INICT subscribers will get the hard copy. Only the offline students will get the hard copy. The online students, you would get the PDF beforehand. You can take a hard copy yourself by taking a printout. Otherwise, you would anyways have the soft copy. So if you are an iPad user, you can make the notes in that as well. Yes, Divya, I personally love biochemistry. Recorded version of what meets do you want to watch? If you're asking about the topic that I was talking about, it's on the Unacademy app. Yes, my favorite subject is also physiology. So basically, because I have read very well in my first year and second year of MBBS, and that's why these are like my favorite subjects. Chalo. For the time being, the INICT course Dave Panda is showing only 2 classes. The entire course will be uploaded. Don't worry on that front. It's not only 2 classes, it's much more than that. All right. So starting with the discussion, DNA replication, the first point to be remembered. It occurs in which direction? Does it occur in the 5 prime, 3 prime or the 3 prime, 5 prime direction? In what direction does it occur? So remember that the replication occurs in the 5 prime, 3 prime direction. So when I say it is DNA replication, basically it is DNA synthesis. So remember that synthesis is basically when I write synthesis, it is S, which is 5. So it goes from 5 prime to 3 prime direction. So one we have is the leading strand, which is synthesized in the continuous fashion. One we have is the lagging strand, which has the Okazaki fragments, which is discontinuous. So it is both continuous and discontinuous. It is semi-conservative. That means the one is the original parent DNA strand, the one is the new strand. So from the 2 strands of DNA, so we have these 2 new DNA copies which are made. So one is the original, one is the new. So that means it is semi-conservative, right? It is more complex in eukaryotes definitely, but the enzymes are almost the same. Origin of replication, that is the ORI region as it is called as. So we have the particular sequences in the genome where the DNA replication begins. In prokaryotes, it is single. Remember that eukaryotes have multiple origin of replication. The DNA replication can start at multiple points. And please remember this point that the promoter, that is actually where the DNA synthesis is starting, uske pehle hota hai promoter, that is which is often upstream. And it is rich in what sequences? Remember that it is 80 rich sequence. Okay, it's an 80 rich sequence. So 80 rich sequence you can remember as promoter is like OT. You can remember it as like 80. Promoter is 80 rich. Promoter or origin of replication is 80 rich. 80 rich is basically your Tata box. That is the 80 is the Tata box or even the CART box. Both of these are present in the promoter region or the ORI where the replication starts. Replication fork is the Y-shaped region, basically where the DNA along the DNA templates where the strands are synthesized. Now coming to the function of the various enzymes in DNA replication, which is that enzyme which causes the unwinding at the replication fork. So remember that helicase, as the term says, helicase breaks the helix, right? So the DNA which is like a helix, it is broken down, unwinding by helicase. Now remember that this is deficient in Bloom syndrome. Okay, so what is the problem in the Bloom syndrome? Bloom syndrome is one of the pro-malignant conditions, right? The gene is the BLM gene. When I write Bloom, remember it is BLM gene. And this helicase is deficient in the Bloom syndrome. Okay, in the Bloom syndrome, the helicase is deficient, right? So remember helicase is halving the DNA, causing the DNA unwinding. Bloom is like bi, dividing into two. So bloom is bi, that is helicase deficiency. What is the function of these single-stranded binding proteins? As the term says, these are the proteins that bind to the single strand of DNA. So when the helicase has caused the unwinding, now we don't want the two strands to stick again together. So we need some proteins which stabilize these single strands of DNA. So basically these prevent the strands from re-annealing or degradation by nucleases. So by binding to the single strand. What do DNA topoisomerase do? Right, we have learned about topoisomerase 1, topoisomerase 2 in the anti-cancer drugs. Topoisomerase, basically they create a break in the helix, right? They break the DNA helix to add or remove the supercoils, right? So basically if there is a lot of overwinding of the DNA, to remove those winding, the coils, this topoisomerase acts. So basically it creates a break in the DNA strand. It could be a single stranded break or a double stranded break. Topoisomerase 1 is single stranded break, and topoisomerase 2 is a double stranded break. Okay, that's a double stranded break. So remember that 1 causes single stranded break and 2 causes double stranded break in the DNA, basically for underwinding or overwinding of the DNA. And look at this point. So which are the drugs which act on topoisomerase 1 and topoisomerase 2? Remember irinotecan, topotecans, the thecans. AN is like ONE1. These are the topoisomerase 1 inhibitors. Etoposide, TO is TWO. It inhibits topoisomerase 2. These are the anti-cancer drugs that we have seen. Remember, where is the irinotecan used? Where is the irinotecan used? Anyone? Irinotecan is used in which cancer? Yes? Irinotecan is used as an anti-cancer drug. In which cancer? It is used in colorectal cancer. Okay, it is used in colorectal cancer. Very good. Colorectal cancer. The FOLFIRI regimen, right? The FOLFIRI regimen, the IRI is the irinotecan. And very, very important point to be remembered for irinotecan is, this is contraindicated in which condition? Irinotecan is contraindicated in which condition? It is contraindicated in Crigler-Najjar syndrome. Why? Because it needs the metabolism by this UDP glucuronyl transferase, which is absent in Crigler-Najjar. Right? And what is the dose limiting side effect with irinotecan? It is diarrhea. It is used for colon cancer. At the same time, the side effect it causes is also the colon related. That means it is diarrhea. Okay, remember these points for irinotecan. So this is topoisomerase 1, topoisomerase 2. Which are the antimicrobials which act by inhibiting topoisomerase? The antimicrobials are fluoroquinolones. Right, A, B, C, D, E, F, G. Fluoroquinolones inhibit gyrase, which is nothing but topoisomerase 2. Right, so these inhibit topoisomerase 2 and topoisomerase 4 in the bacteria. The antimicrobials inhibit in the bacteria. Is this clear with everyone? Right. Now going to the next point. What is the function of primase? What is the function of a primase? Primase, as the term says, does not break the primer. Right. It makes the primer. Remember this point. It is not the primer breaking, like helicase breaking the helix. It is making the primer. What is this primer made up of? Is it a DNA sequence or is it a RNA sequence? Remember that it is a RNA primer. Why do we require RNA primer? For DNA synthesis. To start the replication, the first thing that we need is RNA primer. Right? DNA synthesis requires primer. RNA synthesis does not require primer. Remember, RNA synthesis does not require primer. DNA synthesis requires primer, and this is synthesized by primase. What DNA polymerase do we call it in the eukaryotes, which synthesizes the primer? Which DNA polymerase it is? Is it DNA polymerase alpha, beta, gamma, delta, epsilon? Which one is that? Yes? The first one is the alpha. So remember that primase is basically DNA polymerase alpha. Okay, it is DNA polymerase alpha. Which is the DNA polymerase in the prokaryotes, which is the most processive, that is maximum replication? It is DNA polymerase 3. Okay, it is DNA polymerase 3. Now coming to DNA polymerase 3 and DNA polymerase 1. The DNA polymerase 1, 2, 3 are basically for prokaryotic. For eukaryotes, it is DNA polymerase alpha, beta, gamma, delta, epsilon. Right? Now, remember that this is for prokaryotes. This is the one which elongates the leading strand by adding the deoxynucleotides to the 3' end. Right? It will add at the 3' end. 5', 3' direction may the synthesis occurs. So the new nucleotides are added at the 3' end. Right? And it elongates the lagging strand also until it reaches the primer of the preceding fragment. Now, DNA polymerase 3, DNA polymerase 2 and DNA polymerase 1. Can you tell me what is the difference in between them? DNA polymerase 3, DNA polymerase 2 and DNA polymerase 1. Now, imagine like I've told you the mnemonics for this. Imagine like these are the ranks in the exam. Rank 3, rank 2 and rank 1. Rank 3 being the lowermost. Now, if you keep synthesizing new DNA, that means new stuff in your brain every single day. You are just focusing on looking at new notes, new stuff, new resource. You would get a rank 3. So basically, this is new replication all the time. This is the DNA polymerase, which is the most processive. Right? All the 3 DNA, they do the proofreading. So example that I give, once you give a grand test, once you give an exam, everybody does the proofreading कि मैंने सब questions answer किए हैं कि नहीं किए हैं. Right? It's only if you do the repair, if you see that there are some mistakes and you repair it, then you get the top 2 ranks. So DNA polymerase 1 and 2 are the ones which do the repair. When do you get the topmost rank? That is rank 1. When after giving the grand test, you see that these are my areas where I am lagging and you try to fill those gaps. So DNA polymerase 1 is the one which fills the gaps in the lagging strand. So wherever the lagging strand basically has gaps, these gaps are filled by the DNA polymerase 1. Okay, these are filled by DNA polymerase 1. So this is for the prokaryotic DNA. What is for the eukaryotic DNA? Eukaryotic DNA, remember, is alpha, beta, gamma, delta and epsilon. To begin with the first one, that is the primer synthesis is alpha. Come in the anti-clockwise direction, the reverse circle like this. Epsilon is the leading strand synthesis. Right? Then delta is the lagging strand synthesis. Gamma, yesterday we had seen, is the mitochondrial DNA. Now all the DNA is synthesized. It is the beta which does the repair. Okay, DNA polymerase beta is the one which is predominantly involved in the repair. And what DNA polymerase fills the gaps in the lagging strand? The leading one. That is the DNA polymerase epsilon fills the gaps in the lagging strand. Okay, this is how you remember this. So remember that DNA polymerase 3 basically synthesizing the new DNA in the leading and the lagging. So it has the synthesis function, the 5', 3' direction synthesis. How is the proofreading? The proofreading is the 3', 5', right? We have synthesized in 5', 3' direction. We proofread in the reverse direction, that is 3', 5', exonuclease. Okay, it is 3', 5' exonuclease activity. Is this clear with everyone? Now coming to DNA polymerase 1. Again, it is in prokaryotes. And this is the one which degrades the RNA primer and replaces it with DNA. So the RNA primer in the last, it is to be replaced by DNA. This is done by DNA polymerase 1. Okay? So also remember the additional point of DNA polymerase 1 that it excises the RNA primer with what function? It is the 5', 3' exonuclease activity. Okay, remember this very, very important point. that it is DNA polymerase 1, which has the 5' 3' exonuclease activity by which it removes the RNA primer. The proofreading is by 3' 5'. The RNA primer removal is by 5' 3' exonuclease activity, which is present in DNA polymerase 1. Very, very important. DNA ligase, basically the gaps which are there, it is filled by the DNA polymerase 1, and the sticking happens by DNA ligase. So it joins the Okazaki fragments. Ligase matlab linking the DNA. So what does it do? This DNA ligase is forming what bonds? The phosphodiester bond in the strand. It is not between the two strands, in the same strand. So basically it is forming the phosphodiester bonds. This is something which is very, very important. That is the telomerase. What is telomerase? First point to be remembered that telomerase is present in eukaryotes only, not in prokaryotes. Telomerase is a reverse transcriptase. Now, what is a reverse transcriptase? Transcription means synthesis of RNA from DNA. Right? DNA to RNA is transcription. So reverse transcription is RNA to DNA. That means it is RNA dependent DNA polymerase. We are synthesizing DNA. So it is DNA polymerase. Remember this point as well. It is RNA dependent DNA polymerase, that is reverse transcriptase. It is a DNA polymerase. That means it adds DNA to the 3' end of the chromosome. Basically, it is the one which is not allowing the chromosome to become short. So with every replication, it is preventing the shortening of the chromosome, the loss of the genetic material, by adding the DNA at the 3' end. What sequences is it adding? What is it rich in? Remember that it is TTAGGG. Okay, it is TTAGGE. This is remembered by the mnemonic given here. Telomerase tags for greatness and glory. Okay, so telomerase tags for greatness and glory. This is the sequence that the telomerase has, right? The DNA sequence that the telomerase adds. Okay, so telomerase activity, when is it increased? In the progenitor cells, that is the stem cells, and in cancer, basically, which are proliferating, which are not dying. It is down regulated in aging and progeria, where there is premature aging. In that cases, the telomerase activity goes down, and that is why it is aging, right? So remember that this is a very, very important point. A few important lines for your upcoming exam. Very, very important and frequently asked. Okay. So that is about the telomerase. Now, look at the diagram. You can get an image-based question, and they might ask you that this diagram is representing which enzyme. So first one that you see here is at the replication fork, the unwinding is done by helicase. Okay, helicase does the unwinding. So this is the replication fork which is formed. The leading strand is synthesized towards the fork. The lagging strand is going away from the fork. Okay, the leading strand is continuous. The lagging strand is basically in fragments. Okay, it is in fragments. At the starting of the DNA synthesis is the presence of the primer. Okay, this is the RNA primer. The synthesis occurs in 5' to 3' direction. Okay, so this is the RNA primer. This is the RNA primer which is formed. Now you have DNA polymerase which is forming the DNA, right? These are the topoisomerase. Topoisomerase which produce the breaks in the DNA for underwinding or overwinding. Basically supercoils prevent करने के लिए. To the single strand of the DNA, the proteins which are binding, these are the single-stranded binding protein. That is, they bind to the single strand to prevent the re-annealing or to prevent the degradation. This is single-stranded binding protein. Okay? Is this clear with everyone? So this is what we have in the DNA replication. Now, going to the DNA repair. What are the mechanisms for DNA repair? For double strand and for single strand. This is very, very important. I would tell you that what are the questions that are asked and have been asked. So one is the non-homologous end joining and one is the homologous recombination. So non-homologous end joining basically means that when you have a break in the DNA, like you see here, it's a double-stranded break in the DNA. The non-homologous end joining is basically the joining of these. Okay, the joining of these, basically the strand joining together. Say this strand is basically the non-homologous end joining. So this basically seals the break. Okay, this seals the break. Here you do not require a complementary, a DNA which is homologous to the parent DNA. So remember that non-homologous end joining, basically it brings the two ends of DNA fragments to repair the break. Here the homology is not required, and in the process a part of the DNA may be lost or translocated. And remember that this may be dysfunctional in ataxia telangiectasia is non-homologous end joining defect. Okay, so in ataxia telangiectasia, which comes under your DNA repair defect, there is a defect in non-homologous end joining. Okay? Homologous recombination, basically it requires a homologous DNA. So it requires the homologous DNA, and what happens is, to one of the one strand binds to the other strand. So what is happening is, a strand from the damaged double-stranded DNA is repaired using the complementary strand from the homologous intact DNA. And where do you find this defect of homologous repair? This is defective in Fanconi's anemia and in BRCA1, BRCA2 mutations. Right? This is where this is defective. So homologous recombination defect is seen in Fanconi's and in BRCA1, BRCA2. You can remember this as FA and BR. That is like father and brother. These are like homologous, right? These are like homologous. So basically for a sister, the brother is like a father. They play the homologous role. So remember that father, that is Fanconi's, and brother, that is BRCA. These are homologous recombination defect. Bloom syndrome was what defect? What did we see was the defect in the Bloom syndrome, the BLMG? Bloom is bi matlab helicase defect. Dysfunctional helicase is what we have in Bloom syndrome. Okay, that is what we have in Bloom syndrome. Now going to the other process of DNA repair. When you have a single strand, right, there's a defect in the single strand. So the repair which comes here is nucleotide excision repair, the base excision repair, and the mismatch repair. Can you tell me where does glycosylase come into play? Important question. Glycosylase is required for which of these repair mechanisms? Remember, it is the base excision repair. Okay, it is the base excision repair where glycosylase is functioning. We will have a look at this. Again, this is very, very important. So nucleotide excision repair. So what happens here? You have a sequence in the DNA which contains the damaged bases. We need to remove that sequence. So for that we require endonuclease. Remember that if this is a DNA strand, removing from the middle of the DNA requires endonuclease. Removing from the end of the DNA requires exonuclease. So if in a DNA strand you have defective bases, you will need an endonuclease to remove the damaged bases, right? So once it is removed, we need to synthesize that DNA. So DNA polymerase comes into that place, and then comes the ligase which will do the linking, right? So this repairs the bulky helix distorting lesions like pyrimidine dimers. Okay, when pyrimidine dimers are formed in the single strand itself. And very, very important and very frequently asked, remember that, please bear with the background noise, so kids there around. So remember that this is defective in xeroderma pigmentosum. You get the options like, what is the defect in xeroderma pigmentosa? Is it base excision repair, nucleotide excision repair, right? In that, what do we have is nucleotide excision repair defect. Okay, it is nucleotide excision repair defect. What happens in xeroderma pigmentosum is there is inability to repair the DNA pyrimidine dimers. which is caused by ultraviolet exposure. The patient has photosensitivity and skin cancer. This is the history that is given. This is a frequently asked question, and this occurs in the G1 phase. Okay, this occurs in the G1 phase. Then you have the base excision repair. Base excision repair is you have a glycosylase, which is base specific glycolysis. Glycosylase, it removes the altered base. Let's say a base is altered. Like it is shown in this diagram here. Look at this one. So what is happening here? Deaminated C. Can you tell me what does deaminated C form? Cytosine on deamination. What does it form? It forms uracil. We have seen this in the pyrimidine synthesis. So what is happening? The C is getting replaced by U. DNA ideally should not contain this uracil. So we need to remove this. So what happens? This glycosylase removes the uracil. Right? Endonuclease and lyase, they remove the background and the glycosylase just removes the base. So base is removed by glycosylase, the backbone is removed by the endonuclease and the lyase. And what happens? Then comes the DNA polymerase, which is getting the right base here. Okay, which is getting the right base here. So this is what is base excision repair. Remember that the glycosylase creates the AP site, that is apyrimidinic site, because you are removing the base, and then the nucleotides are removed by endonuclease, right? And then you have the lyase, which cleaves the end. Then comes the DNA polymerase beta, which fills the gap, and you have DNA ligase, which seals it. So all of this is required for base excision repair. First is the glycosylase, right? First is the glycosylase, then you have the endonuclease, then you have the lyase, then you have the polymerase beta, and then you have the ligase. This is what is given here, the sequence. GEL, please. What does it stand for? Glycosylase, endonuclease, lyase, polymerase, and ligase. All of these are required for base excision repair. This occurs throughout the cell cycle, and remember that this is important in repair of spontaneous or toxic deamination. As we saw, deamination of cytosine forms uracil, तो उसको निकालने के लिए. So remember, for deamination, it is base excision repair. Okay, it is base excision repair. Coming to the next one, that is the mismatch repair. Very, very important. This is defective in HNPCC. Very, very frequently asked. In HNPCC, it is the mismatch repair defect, also called as the Lynch syndrome. Okay, it is also called as Lynch syndrome. And this occurs predominantly in the S phase of the cell cycle. So the mismatched nucleotides, they are removed. Like, look at this diagram here. So here this is the mismatching. The G is mismatched to A. The G should be binding to C. So what is happening is this mismatch segment is removed and then the proper segment is filled in. This is mismatch repair. Look at the nucleotide excision repair. When you have the pyrimidine dimers, which are formed on a single strand, the thymidine is binding to thymidine. So this is what is forming the pyrimidine dimers, which is because of the ultraviolet exposure. Okay, this is because of the ultraviolet exposure leads to pyrimidine dimers. Very, very important. The endonuclease removes the segment, right? And then you have the newly removed segment. So I hope everybody is clear with the points that we have learned here. What did we learn here in this DNA repair? In the DNA repair, we have the double strand repair, the single strand repair. Double strand repair is the non-homologous end joining and the homologous recombination. Non-homologous end joining is basically where this and this, they join together, the seal happens. This is defective in ataxia telangiectasia. Homologous is defective in, homologous is brother, that is BRCA, and father, that is Fanconi anemias. Homologous requires the complementary intact DNA. Okay, it requires a complementary intact DNA. In the single-stranded DNA repair, what do we have? We have the nucleotide excision repair, the base excision repair, and we have the mismatch repair. Nucleotide excision repair is defective in xeroderma pigmentosum. This is required for pyrimidine dimer formation, which is basically because of UV light. UV light creates that thymidine dimers. Base excision repair requires glycosylase, right? Gel please is the sequence: glycosylase, endonuclease, lyase, polymerase, and ligase. This takes place throughout the cell cycle, and this is important for deamination reactions, right? Spontaneous deamination जब होता है तो उसको remove करने के लिए. Mismatch repair is defective in HNPCC, also called as the Lynch syndrome. Okay, that is Lynch syndrome or HNPCC. Right. So this is what we have here in basically your DNA repair. Okay, this is about the DNA repair. Now, quickly coming to the DNA mutations. Now, DNA mutations could be single nucleotide substitution or other mutations. Single nucleotide, matlab at one point itself. That means basically these are point mutations. Okay, these are point mutations. Now, what happens in this point mutation? Basically, single nucleotide is replaced. So, it might result in silent mutation, missense mutation, or nonsense mutation. Silent mutation means even if the nucleotide is changed, but the amino acid synthesized is the same. So it is the same amino acid. Which position is generally involved? It is the third position, because we know that is where the change can occur without any change in amino acid, right? The wobble hypothesis. We saw today the wobble hypothesis is for degeneracy or for redundancy, right? Next, we have missense, where it's a different amino acid now which is synthesized, right? So, example, this has been asked in your previous NICT exam. Sickle cell disease is an example of point mutation, missense mutation, because what happens in sickle cell anemia is instead of glutamic acid, there is valine at the beta 6 position, right? At beta 6, it is glutamate replaced by valine. So one amino acid replaced by another amino acid. It's a missense mutation. Nonsense is a stop codon, where the codon is changed to a stop codon, right? So stop the nonsense. So remember, stop codon wala is the nonsense. Which are the stop codons? The UGA, the UAA, and the UAG. So this stops the protein synthesis early. What is transition and what is transversion in the DNA mutation? Transition matlab replacing purine with purine. Transversion matlab the entire version is changed. Purine is replaced by pyrimidine or pyrimidine by purine. That is transversion. Okay, that is transversion. Now, what is a frame shift mutation? Frame shift mutation could be a insertion or a deletion, which is not in the multiples of 3. So basically, it shifts the frame. Because the frame is read in 3-3 ka multiples, the codons 3-3. So the removal which is not in the multiples of 3 or the insertion which is not in the multiple of 3, which changes the frame, that is called as frame shift mutation. So remember that deletion or insertion of any number of nucleotides not divisible by 3, which leads to misreading, that is what is frame shift. And very important, remember DMD is an example of frame shift mutation. The Duchenne muscular dystrophy is frame shift. Tay-Sachs cystic fibrosis is also frame shift. Okay, that is frame shift mutation. Very important, and this is asked in the exam. DMD is an example of frame shift mutation. Next, we have the splice site mutation. The splicing of the mRNA occurs. What is splicing? Removal of introns. Introns are the interfering sequences. They are not basically expressed. So, if the splice site mutation, we have retained the intron in the mRNA. So, the intron is not removed. That is the splice site mutation. Right? Examples, again, you have Gaucher's disease, Marfan syndrome, which is splice site mutation. So, remember that single site point mutation could be silent, missense, or nonsense. Same amino acid, silent. Different amino acid, missense. Stop codon, it is nonsense. Okay, it is nonsense. The U wale codons, are your stop codons, the UGA, the UAA, and the UAG. Frame shift mutation is when it is not in the multiples of 3, the deletion or the insertion. DMD is an example of that. Even cystic fibrosis. Splice site mutation is when the introns are not removed. That is the splice site mutation. Okay, that is the splice site mutation. Correct? So this is about the today's session in the fast episode 6. Like I said, we will have the continuation of these sessions, the rest of the biochemistry. We are going to see tomorrow at 11 a.m. and at 2 p.m. This is going to be on the Unacademy app as a special class, right? That's a free live class. So that would be fast episode 7 and fast episode 8. Again, it would be related to biochemistry. Okay, again, it would be related to biochemistry, right? So that was about this session, guys. And where are the rest of the videos? And the rest of the videos links you would find in the Telegram group. I will try to combine all of them and give you like in one message. A lot of students have been asking where are the rest of the links. I'll give you at one place maybe tonight. Lac operon, Rock Anonymous, we'll do it tomorrow, right? In the tomorrow's class, we will do the lac operon wala part. Okay, so whatever we have learned today, highlight it, write it down in your notes because this is very, very important. The DNA repair defects, the DNA mutations, the DNA replication, all of this is very, very important and frequently asked in your exam. So I hope you have enjoyed this and you have learned out of it. And the first aid is not as difficult as it seems, right? Thank you so much and goodbye. Take care and keep studying, keep revising, and keep winning. Thank you so much.